From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 12, recorded on November 10th, 2020. I'm Vincent Racaniello, and you're listening to the podcast about the nervous system. Joining me today from New York, Ori Lieberman.
Hi, Vincent. How are you?
We have reached the key episode number 12. Most podcasts don't get past 12 episodes.
I actually had a dream. I had a dream about that the other night where someone was like, "Oh, I heard this is episode 12." And I was like, "Oh, how'd you know? That's really strange." Then I woke up. So.
Wait, so why is this the magic number?
On average, most podcasts don't get past about 12 episodes.
Huh. All right.
So
Did not know that.
If you get past that, you're probably going to be okay. So we made it. It took us a year, I think, right?
Yeah.
Pretty much, yeah.
Also joining us from Salt Lake City, Jason Shepard.
Hey, Vincent. Hi, Ori.
Hey, Jason.
You got snow out there?
We do. It dumped a lot this weekend, even in the valley. So winter is here. But, so Friday I left to go camping in the desert. And it was 75 degrees in Salt Lake. 75. Come back and it's like 28.
Wow. It's quite a change.
So, yeah. Winter storm came through.
And we had a new president, kind of.
Well, I thought I could finally feel some optimism about where we're going in this country. And now, yeah, they're doing everything they can to, destroy the the very institutions that they're supposed to uphold.
Unbelievable.
Let's not get into the politics, right? But yeah.
Well, at least we have a COVID-19 advisory committee, right?
For the president-elect, yeah, right.
Yeah, whether they actually get to work, who knows? Yeah.
Yeah.
All right, well, we have a couple of very cool papers tonight, and I will hand it over to Ori, and we'll learn all about working memory, right?
Yes.
Yeah, it's a different kind of memory from the one previous couple episodes ago.
So working memory, if you can think of, I was trying to think of a good analogy earlier, and I thought that it might be easier just to kind of use a behavioral task that tests working memory. And so if you give a cue, so for example, let's say a light turns green, you have to wait to make a choice based on that cue. You have to hold you have to remember that the light turned green for that period of time, which is called the delay, before making the choice, and that's working memory. So I may or may not have butchered that, but
no, that was right. But let me simplify it even more than that. So It's basically like if I ask you to memorize a telephone number, and you have it in your mind for a few seconds. And then if I come in and interrupt you while you're trying to memorize the number that's gone. So that working memory is sort of that sort of few seconds where you're holding it. I like to think of it as as the brain's RAM, right? It's the memory that's never actually written to the hard drive, as just sticking around for a few seconds to use it. And then if you don't use it, it's out.
And if you can imagine, this is really important in terms of tasks that require attention and just in everyday life. And so obviously understanding the neural basis of this could really be useful.
Totally.
Go ahead.
No, that's exactly right. I mean, this is the bread and butter of cognition. So memory, of course, long-term memory makes us able to access information at any time. But in real daily existence, to do almost anything, we need working memory to know where we are in the world, to know how to make a decision, for just about any task that we're doing.
Is there also some kind of intermediate length as well, besides short and long? Yeah.
So there's working memory, which is sort of this really very short transient thing. There's short-term memory, where if I ask you to memorize the telephone number and I come back a few minutes later and say, hey, what's the telephone number? If you can remember it, then that's short-term memory. If I come back a day later and I say, do you remember the telephone number you memorized yesterday? And then you can, then it's converted into that long-term memory.
Yeah.
That's sort of the distinction.
And long-term memory probably requires repetition, right? It's like if you use that phone number over and over, eventually you're going to remember it for a long time, right?
Exactly.
Right.
Exactly. So the more, repetition is key. But there's still some things like, what we talked about earlier, where if you get a shock, if I shock you, or if you have a traumatic event happen, you don't need that to happen twice to remember it. It's the brain is wired up to remember that. So there are other sort of memories these are so-called flashbulb memories.
Are there people with deficits in specifically working memory, Ori?
So that's what I was going to bring up. So working memory and attention is of course impaired, for example, in attention in like ADD, but also something that I've been seeing a lot in the hospital over the last bit of time that I've been there is delirium, where a person comes into the hospital and has an acute change in cognition. And a lot of times that's in the domain of working memory or attention. And so some things that we do to test for attention, and if you have ever been to the emergency room after hitting your head skiing, for example, they'll say, can you spell world backwards? And that's an attentional task that a lot of people can't do when they're delirious or have this kind of acute change in cognition. And that's different than our tasks for short-term memory, where we'd say, remember 3 words, and I'll ask you about them in 5 minutes or in 20 minutes for longer-term memory.
You also see similar kinds of deficits in working memory after concussion. So that's why there's like the tests for concussion are, sort of like, how can you fail them? But your brain cannot process some really basic stuff after traumatic brain injury. And then there's also some more subtle defects in psychiatric disorders like schizophrenia, where attention, there's some subtle defects you can tease out with attention.
You know what I have a lot of trouble with, and this is working memory. So you go to a web page and you have a number, and then you go to another tab on your browser and you have to type it in somewhere, and you can't see the original one. So I can't even remember 8 digits. By the time I get to the 6th, I'm like, shit, what's the other 2? And I have to go back.
Well, that's actually, this is a bit of an aside, but this is a fascinating to me aspect of working memory and numbers. So there's a reason why most telephone numbers are 6 or 7 digits, because it turns out that the capacity for almost everyone, every human, is 6 plus or minus 2 digits. Cool. To keep in mind. So you're totally fine. This is normal. But what's fascinating is that it's tied into how we use language. So there's some studies in the Amazon or indigenous populations that don't have numbers for more than 2, and it turns out in that case, working memory is altered so that they can't actually hold less objects in their working memory than in other cultures. And so it sort of goes back to this idea of how thought can constrain cognition. But this idea that you can only hold maximally 6 or so objects or numbers or whatever in your working memory seems to hold across all human populations, which is fascinating.
Interesting.
Yeah.
Interesting. Cool stuff. Okay. Go ahead, Ori. Sorry.
No. So in this paper, they're trying to understand the neural basis of working memory. And there has been a lot of work done on how working memory is stored and kind of maintained in the brain. And most of this work has been done in a mix of monkeys and mouse models and humans, have shown that the prefrontal cortex, so the very front of the brain, and parts of the thalamus are important in working memory. And the way that they think that the prefrontal cortex, which I'll abbreviate as PFC, kind of switch back and forth if I remember to say prefrontal cortex or not. So the way that the PFC encodes working memory is by changes in firing patterns in the pyramidal neurons or the projection neurons of the deeper layers of the cortex. And these changes in firing patterns tend to correlate with the length of the delay. So for example, in a behavioral task, if you say you have to hold one piece of information for 1 second, these neurons will sometimes change their firing rate for exactly 1 second before that information is then used for a choice in the task, for example. And these are, so for the last 30 years, neuroscientists have tried to understand how these firing patterns, what the mechanism of change in these firing patterns are. And initially it was thought that this could be driven intrinsically. So a change in the ion channels in one particular neuron could lead to a pause or transient change in firing rate. And then there was kind of this wave of groups that suggested that it was local circuitry. So in the cortex, there are these like highly collateralized axon networks between these excitatory pyramidal neurons and local inhibitory interneurons, which, as you can imagine, if there's kind of this feed-forward loop, could generate longer periods of pauses in firing rates. And more recently, in the last 3 or 4 years, there has been this idea that other brain regions outside the cortex, in particular the mediodorsal nucleus of the thalamus, is important for generating these pauses or changes in firing rates.
And just to interrupt, but the thalamus is thought to be basically like the relay station of the brain. So it's a subcortical, it's a nucleus in a large nucleus that receives input from all over the cortex and then relays that information to other parts of the cortex. And so it's kind of been understudied because of that, because the concept was that it was mostly just there to act as a station for information to go through, and in itself is not really participating in interesting things like working memory per se.
And in the thalamus, the nice thing about this idea is that in the thalamus, many of the neurons rhythmically fire. So it actually could explain how you could generate rhythmic activity or transient changes in firing if you imagine that the thalamus then is disinhibited, fires rhythmically for a period of time, and then changes the cortical firing.
Right. And then what's fascinating here is that, so the working memory, the timing is critical. And so basically think of these looping events, these of specific circuits have a clock somehow, but it's not been clear where that, what determines the clock. So how does that get timed with the behavior or the output of the task?
And so a lot of the evidence for this idea of kind of corticothalamic networks in working memory has come from just basic neuroscience experiments where, for example, if you inhibit the thalamus during the delay period, you can essentially make animals forget what the initial information was. But in this paper, which I'll mention in a second, they took a completely different approach, which I think was super elegant. So what we'll discuss is a paper called The Thalamic Orphan Receptor Drives Variability in Short-Term Memory. It was published in Cell. The first author is Quang-Fu Xiao, and the last author is Priyambada Rajasupati, and they're from Rockefeller University.
So local, kind of.
Yeah, we're too New York focused, I think, sometimes. So
I'm here.
So yeah, that's we have our token Western man. Yeah. So what this group did was leverage two groups of outbred mice to try to understand what the genetic basis for working memory is. And let's just take a step back and think about what outbred mice are. So in most of our experiments in the lab, we tried to minimize all of the variables that could interfere with the one question that we're asking. And one way that we as scientists do this is by making purely genetically pure populations of mice. So if you can imagine, you have a male and a female mouse, you breed them together, and then you take the next generation and you breed them back. So you can essentially have genetically identical mice. So when you compare two conditions, you're comparing them on a background that's exactly identical. And this actually, from a historical perspective, is really cool because this came out of mouse breeding in Europe and kind of in the royal societies where people bred mice for specific characteristics, and then they became kind of like these genetically kind of unique populations.
So like dogs.
So like dogs, exactly, or cats. Yeah, well, less so cats, but but the downside of these experiments is that you lose normal variability in mice, which you could take advantage of to understand differences in their behavior. And so what they're kind of over the last bit of time, there's been a push to bring back this sort of discovery into science by generating populations of mice that are diverse genetically, characterizing behavioral or biochemical differences, and then going back and looking at the genetic differences to try to understand the genetic basis for these differences. And this is done in humans all the time with genome-wide association studies or GWAS studies. So if you think about it in a simple way, you could imagine just doing GWAS studies on mice. And so what this group did was they took a population of outbred mice, so genetically diverse mice, and they phenotyped them in two different behavioral tasks that test working memory. So in the first task, it's called the spontaneous alternation task, and I find this to be a very interesting task because it's a little bit strange to me. So if you put a mouse in a 3-armed apparatus, so there's a center, and then they can then choose to go out one arm, a second arm, or a third arm, and you let them explore, they will very rarely will they go back to the arm that they just explored in. So if you imagine that there are arms A, B, and C, if they come from A, they go to B, they very rarely go back to A. They'll usually go to C. And the idea is that this is kind of taking advantage of mouse's innate interest in exploration, but they also have to remember where they were before, what the last arm they were in were. And the advantage in doing this sort of long or large scale behavioral screen is that it's a really easy task. So you don't have to train the mice to remember one thing and have a period of behavioral sessions where they learn this thing and then you test their memory of it. You essentially can just take naive mice, throw them in a behavioral apparatus and get your readout.
Yeah, and so just interject. I mean, this sort of strategy of looking for genes that correlate with behaviors has previously been very difficult in mice because, one, you needed many mice and they're expensive, and two, you needed this sort of easy behavior. And then the third thing that you'll get into is the cost of sequencing. You know, it's very easy to sequence the genomes now of whole animals, or even, basically doing the single cell sequencing and getting to the point where you can sequence just one cell at a time. So it's this is a fruition of a bunch of different technologies that are coming together in one study, really.
And the other really cool aspect of this is that in some way this kind of set of experiments is agnostic to how working memory actually works, because you can imagine that you identify genes that are in one part of the brain and another part of the brain, but you're not biasing yourself by existing theories of what is the basis of working memory. And it's actually a really powerful, we'll get to what they show, but it kind of really powerfully emphasizes one of those theories that we discussed a few minutes ago in being the kind of the basis for working memory. And so what they do is they phenotype almost 200 mice from this group. And they find that there's a large variability that could be that was heritable, essentially. So you can imagine that, of course, if you take any population of mice, there will be some variability, just like if you took any population of humans, there will be variability in any one of our characteristics, like height, for example. But the idea is that they're looking for a characteristic that is heritable, so should have some genetic basis. And they show using some math that is very much too advanced for me that there is heritability in the working memory task. And then they do a number of controls, which I thought were interesting, and we can kind of talk about it separately, but they showed essentially that there was no correlation between performance in the working memory task and like a wide range of other characteristics of the mice. So, for example, like motor function, exploration, the weight, or what they call body physique, or sensation. And I mean, to me, this was very interesting because, of course, you can imagine that performance in this task should be dependent on the exploratory drive or the motivation to explore these different arms. So it's kind of cool that they found that there was some unique aspect of it that was captured in the genetics. And then they sequenced and genotyped these mice. So, I mean, it's truly remarkable. So they essentially did QTL mapping of these mice, of 193 mice. And what they found was a region on chromosome 5 that was highly correlated with performance in working memory. I don't know if you guys want to go into more of kind of the genetic analysis, because I think it's a little into the weeds, but
yeah, I think it's, yeah, I think it's not the most interesting part of this, but you know, I guess one of the things that I'm was surprised by is, you know, they've got a couple of hits here, and they've got this one massive one, but you know, I guess what I didn't see discussed was sort of the there's a genetic component to this, but there's also always some aspect of the parents, the influence of the parents on offspring. So if if you have a bad mom, potentially you would have effects on the offspring, and they didn't control for that, I don't think, but they still see this large genetic. I mean, so it's really a big hit that they've got.
It's also worth pointing out that this locus on chromosome 5 explains 17% of the variance in these memory tasks, but there are lots of smaller peaks. They say we need more mice to resolve those, and there may be other genes involved, right? So this is just one that has a match.
Oh, totally. I mean, and this cutoff points are arbitrary. Like they're saying, well, we're just going to call this, statistic, the statistics that they use to make a cutoff for what could be hit and what is not is arbitrary in the end.
And of course, if they had genotyped 1,000 mice, they would find more genes with a smaller effect size.
Right.
Yeah. So these are always correlated. So they then do a smart thing, which is they name this locus the smart 1 locus. And they look within the smart 1 locus, and they find that there are different haplotypes. So a haplotype is essentially like a set of mutations that are present in one area of a chromosome that always segregate together. And this haplotype, which they found in a subset of mice called the cast mice, were associated with a higher performance relative to the haplotype that was seen in C57 black 6 mice, which are generally the standard laboratory mice that most labs use for neuroscience. And so this actually gave them a bit of a trick, because now they could use C57 black 6 mice as one of their quote, their dumb cohort, and then their smart 1 cast mice as their smart cohort, and try to find what the gene was, or what the specific mutation was that was associated with improved working memory. Then they also use and this is a bit of a side point, but they also used a separate set of outbred mice called the collaborative cross mice. And these are really nice because they are defined populations of individuals. So it's the same set of parents that are then crossed together, but instead of then having a random set of offspring that you have to sequence individually in your experiment, these collaborative cross parents have been bred, and then there are a subset of new genetically homogeneous populations that are the offspring. So it's actually a much cheaper and kind of less resource expensive way to do these type of outbred genetics. And they also saw that this cast haplotype in the smart 1 locus was correlated with better performance and working memory in offspring in the collaborative cross that had this haplotype.
So we have these mice, these collaborative cross mice in our lab. You may remember Ori talking to Amy about this. So we're looking for mice that can be infected with a virus. So the virus does not infect C57 black or other, inbred lines. So we're hoping in these outbred lines to find some that can be infected. Then we'll map the genes that allow them to be infected, very much like they're doing here, except for a virus infection.
Yeah.
Cool.
It's a super powerful technique. It's
very
really
expensive, as you said.
Yeah.
I mean, if you want to buy these mice, they're $200 each.
Jeez.
Wow.
Not to mention the cage costs and everything else.
Someone gave us 11, I think, lines, breeding pairs for 11 lines, which is thousands of dollars of mice. Now we can breed them and you don't have to buy them, but just to get them initially, it's a huge expense. Mouse studies are expensive. Yeah, and the housing too.
Yep. Tell me about it.
Got to pay for the mouse hotel. Yeah.
Yeah, I mean, I think we spend probably like $20,000 a month on mice.
Wow.
Yeah.
How many mice do you have? Thousands?
Lots.
Holy cow.
The lab that I got my PhD in was spending you said 20,000 a month? They were spending between 150 and 200,000 a year, so that's about the same.
It's about the same.
So, yeah, similar. Yeah.
I mean, it fluctuates between 10 and 20. But anyway, yeah, this is most of what our costs are for our science is animal.
More expensive than people, huh?
Well, I mean, the salaries are still the other expense.
I wouldn't mind making $10,000 a month, though. That would be great.
That's true.
Yeah. So the next part of the paper is that they wanted to nail down within this region of chromosome 5 what the direct mutation was that was conferring improvements in working memory. And you can imagine that there's a couple of different ways that this could happen. So this could be like a switch in a codon within a particular protein. So you could have, let's say, for example, an ion channel that conducts ions more efficiently. It could be a premature stop codon. So you could have a loss of function mutation in a particular protein. Or they could be intergenic SNPs, single nucleotide polymorphisms, that affect gene expression of other genes in this locus, or frankly, in other parts of the genome. And what they found was that the majority of SNPs in the Cas haplotype of chromosome 5 were in intergenic regions. And of course, as someone who wants a simple story, this I'm sure was a bit disappointing to them, because it would have been really nice to have a single mutation in one gene, and then you can characterize the gene, and then you have your cell paper. So, but what they did was they took a very interesting approach to this. So they hypothesized that a SNP in the intergenic region, so the area between genes, which doesn't directly code for a protein, would be affecting expression of these proteins, expression of other genes in the region of this chromosome. And if you did RNA sequencing of areas of the brain that were involved in working memory from these two different populations of mice, you could then do like differential expression analysis, like we've talked about previously with RNA-seq, and see genes that are differentially expressed and could therefore be mediating the effect on working memory without a true mutation in that gene itself.
Yeah, so this is where modern technologies really come in, and I mean, it's still. I'm not a huge fan of a lot of these RNA sequencing papers because it's basically just saying, well, we can do this, so we're going to do it. I love this paper because they use it for a really specific purpose that you could never do anywhere in any other way. Because like usually if you had a mutation or a change in a non-coding part of the genome, you're just like, oh, well, we think this is going to affect expression of other things, but how do we know? How can we tell? And so this is a real good use of RNA sequencing.
And the way they designed the experiment is very interesting because they took prefrontal cortex, thalamus, medial dorsal thalamus, and hippocampus for their sequencing studies. And in some ways, this is a unbiased approach to figure out where the gene expression differences would be the greatest between these two strains of mice. And what they found was that there were bigger changes in gene expression in the thalamus than in the PFC. which is really, the PFC is thought to be the home of working memory in the brain. So that was just kind of some evidence in favor of the thalamus being the critical player in working memory. So then to hone down on the specific gene, they essentially created a set of criteria that, again, are somewhat arbitrary. But what they said was they would look for genes that were differentially expressed between high and low performers on the working memory task and had robust expression. So as you can imagine in these RNA sequencing experiments, if there's a gene that has very low expression, let's say there are 10 reads, if you have a 80% reduction, there could be 2 reads, but based on the noise of the assay, that could also go to 0, and then you don't really know what the true reduction in expression is. So if you have, the more reads you have, the more confidence you have that there's that you're capturing the true magnitude of the difference in expression. And what they found were 4 genes, so 3 that were differentially expressed in the thalamus, and 1 that was differentially expressed in the PFC. And then they did a series of experiments with each of these genes. So again, taking advantage of some of the technology that we've talked about in previous episodes. So they generate adeno-associated viruses that either overexpress these genes of interest or express inhibitory, so RNAi constructs to reduce the endogenous expression of these genes. And they inject them into either the mediodorsal thalamus or the prefrontal cortex. And then they did their behavioral assays, which they had worked out earlier in the paper. So either spontaneous alternation or another task, which we won't go into, and I didn't bring up before, but that showed similar strain differences in performance. And what they found was the most robust effects for a protein called GPR12, which is a surface protein, so what they call an orphan receptor, which I'll explain in a second, that had really previously not been very well characterized. So an orphan receptor is a membrane protein, and they're called orphans because it's unclear what the ligand that binds them is. So, for example, if you think about the sympathetic nervous system, or you think of adrenaline, so the adrenergic receptors are not orphan receptors because we know that adrenaline binds to them and induces signaling. But in this case, we don't know what is binding to this protein. And that also makes it an interesting kind of scientific question, because receptors then are easily druggable because they're on the cell surface. You can theoretically generate ligands that can bias their signaling, and make it a very interesting way to, if you imagine someday in the future, you could have cognitive enhancement by targeting this protein.
This is also like, the holy grail of this kind of screen, because you could do all of this work, and it's a lot of, and as you said, there's a lot of mice, a lot of expense, and you find it's like this ion channel that everyone knew about already. Mm-hmm. And then you're like, oh, okay, well, at least we know that the screen worked, but what new did we, what did we find anything new? Whereas here, not only you're discovering a new receptor that could be involved, but it's also not even in the same brain region that you expected it to be.
This is the dream. Yeah, this is awesome.
What I think is also cool is that they overproduce this, and it makes them better.
Well, let's not jump the gun.
Oh.
But that's exactly. I mean, this is a real amazing tour de force, because they go from identifying the receptor through this massive screen all the way to like the functional experiment.
It's really remarkable that you can put this virus AAV in with RNAi and knock it down sufficiently to get an effect. It's just those.
Well, it shows the power overexpressing a membrane protein.
Yes.
And actually having it targeted appropriately. It's pretty crazy, right?
Yeah. I mean, you're just sticking this in the brain and it works, and it's just the power of vectors that's part of it, right?
Yep. Yeah, they're efficient.
Yeah. So this receptor is a GPCR, so G protein coupled receptor, which is a family of membrane receptors that are widely available, or like widely used in biology, and they then have activate signaling cascades that lead to changes in downstream function of different pathways. So, for example, changes in calcium signaling, changes in protein kinases, changes in second messengers like cyclic AMP. And what they hypothesized was that GPR12 could actually be affecting some of these signal transduction pathways more or less efficiently, or could be differentially affecting the signal transduction pathways depending on its expression level. So what they do is they use some heterologous cell lines, and they show that overexpression of GPR12 potentiates the effect of glutamate on calcium signaling. And as we've talked about in previous episodes, intracellular calcium is really important for changes in synaptic plasticity, for action potential, for synaptic vesicle release, and for many things for gene expression. So you could kind of tell yourself a very elegant story of how GPR12 can be affecting neuronal function. And the other thing that they do, which I found really nice, although I think it's difficult to show this in the way that they did, was they looked at where this protein is expressed in the neurons, and they found that it's expressed along the thalamocortical axons, so that you can imagine that this is changing kind of the firing properties of these thalamic cells. So all of this comes together really nicely to tell a story that GPR12 in these thalamic neurons is important for working memory. And the last part of the paper goes back to some of the stuff we talked about earlier in our discussion of how changes in firing rates in the prefrontal cortex can mediate working memory. And some of this work has focused on oscillations and kind of synchrony between cortical firing rates and thalamic firing rates, with the idea being that if you have high coupling between these two brain regions, you can promote working memory, and vice versa. So one really nice way to show the role for GPR12 and this haplotype in working memory would be to see whether synchrony between thalamic neurons and cortical neurons is disrupted or changed when the expression is different. And they do this using calcium imaging, which we've also discussed. So they have mice that are freely moving. They have them in a behavioral task, and then they can image genetically defined populations of cells either in the thalamus or the cortex. And they can see that at different times in the task, there's different levels of synchrony between cortical and thalamic populations of neurons, and that this is modified by GPR12 expression level, and also in the different populations of mice from the original screen. So this brings us, it goes from a behavior to a gene all the way back to to maybe a cell biological mechanism back to a neural circuit mechanism, and kind of really, it's just a tour de force paper.
It's just astounding how much work that went into this one paper. It's really, it just highlights, unfortunately, where neuroscience has gone, where to get some of these high impact papers, you have to do something like this. That's probably, we're talking about a couple of $1 million over the whole scale of the study, and goes from behavior to gene sequencing back to behavior, and then functional studies. It's just amazing.
How many years do you think this took to do?
I would say 3 to 5 years.
Oh, okay.
I don't know. I mean, I don't know these groups, but I think they're relatively new PIs. I'm not quite sure.
Yeah, I think Priya gave a talk at Columbia maybe like a year ago, and I think she said she'd had her lab for like 5 or 7 years. So I can imagine. I mean, just in terms of the mouse breeding and the number of generations that you have to have it, I mean, it's years.
Interesting. It's interesting. This is all supported by NIH grants. I was thinking maybe there's some Hughes here because of the big bucks, but all NIH.
Well, I think part of it is that because they were new investigators, they probably had nice startup packages.
Yeah, sure.
And I think this is, I mean, to me, having just gone through this I guess I'm not a new PI anymore.
Nope.
With tenure, but I think this is the great thing about having a great system in America, where when you start out, you get this massive package to start running your, launch your lab, and the world's your oyster. And there's not a lot of times, I feel like, in your career where you're starting from scratch and you're in an environment that says, okay, well, go for gold. Here's your money, and you can sort of do these high risk kind of endeavors. And of course, the other time when you could do it is if you get HHMI, but that's obviously not going to happen for most of us.
You know what my massive startup package was in 1982? $50,000. Now you get 5-6 million easily, depending where you go. 1 or 2 million.
That's a lot.
Yeah.
1 or 2 million for sure.
And my salary was less than that.
In the 80s, well, what's that worth now?
I don't know. I think it's doubled probably, right?
Yeah.
Anyway, this paper took a year to get published. It was submitted about a year ago, October 2019. Took a year. They made him do probably a lot of experiments.
Yeah, and that's actually, I think, relatively average for cell.
Kind of crazy, I think.
Yeah.
Anyway.
No, I think it's becoming an issue, because it does mean that these high impact papers are so resource intensive that it kind of eliminates a lot of people that don't have the same resources. I mean, there's a reason why there's a concentration of these papers from specific institutions.
So Ori, they don't really know how this gene works, right?
Sure.
They have some ideas about calcium and so forth, but that should be figured out, right? How to actually...
Yeah, I mean, that sounds like the basis for the next R1 and HHMI and all. Like, I mean, this is the start of a long story. So I just before we wrap up with this paper, I think that there were a couple things that would have been really cool to see. So one would have been to see if there are genotype variation in this gene in humans. So they make a comment that there's like pretty good homology between this gene in mice and in humans, but I'm sure that there are genomic databases out there that have that. Although I guess it kind of ethically it's complicated, because then you're saying that there's some correlation between genetic background and intellect or working memory. Or at least if there's susceptibility to diseases, that would be interesting also. And then the second thing that I thought would have been really cool, which is more of an experimental question, is that I felt like they were missing an experiment where they overexpressed GPR12 in the mice that were already expressing the higher expressing haplotype. And that I think could give us information about how it's working, whether there's essentially a ceiling effect on working memory, or if you overexpress this gene in everyone, you could improve working memory. It seems like they had almost everything else, and it was just something that I missed in these figures. But I guess I'm reviewer number 3. Yeah.
Well, I mean, there's so many launching points from this paper that if I think for either one of these labs investigators, they should be feeling pretty good about it. I mean, even at the end where they say basically that this whole endeavor is a proof of principle that this kind of approach works. And so one can imagine that you could then, just use a similar approach for almost any phenotype or any behavior that you want to look at within reason, of course, that because you want an assay that's fairly straightforward and you don't have to that doesn't take months to train a mouse, because you're going to have to do lots of them. But in terms of proof of principle, it's really pretty cool. And then Vincent, as you said, there's also these other potential hits that they could probably follow up on as well.
Mm-hmm.
Yep. Good stuff. Nice paper. Very nice. Okay. we had a second manuscript we want to just touch on briefly because it combines viruses and the nervous system, and what could be better, right? What could be better would be a virus that affects working memory. That would be cool.
Or something that's virus-like that affects working memory.
Hey, like your particles, right?
That's right.
I mean, they could be involved in working memory, right?
Well, we have some evidence that the knockout of this gene has some prefrontal cortex activity differences, but it's not clear.
Your gene is called ARC, right?
Called ARC, yeah.
Okay. Anyway, this is a paper from Stan Perlman's lab at the University of Iowa. Stan Perlman is a coronavirus immunologist, and we had him on TWiV some time ago. 4 first co-authors, Zhang, Wang, Li, and Verma. COVID-19 treatments and pathogenesis, including anosmia and K18 human ACE2 mice. I really want to focus on the anosmia because they test convalescent plasma, but as far as I can tell, convalescent plasma, it doesn't work. And monoclonals are replacing it now anyway.
Right.
So this is interesting. This transgenic mouse, it's got the human ACE2 gene, which of course encodes the receptor for SARS-CoV-2. These were made during the SARS-1 outbreak because it was found that SARS-1 back in 2003 had ACE2 as a receptor. And Perlman told us about a year ago they were about to get rid of all these mice because they weren't using them.
Wow.
And they had sent them to be frozen, and then SARS-CoV-2 broke out, and he called wherever he was sending them, Jackson Lab or something. He said, no, take them back. We want them back. So they have them. And it's K18 is a keratin promoter, cytokeratin. So you get expression in epithelial cells. But it's pretty broad. I guess ACE2 in humans is also broad, but it's different from the normal ACE2. Anyway, I remember when we talked to Perlman, he said those mice are not so great because they develop CNS disease, and that doesn't kill SARS-infected patients, the first SARS, right? But now we're seeing with SARS-CoV-2 more neurological involvement, not necessarily virus reproduction, but some symptoms for sure. I mean, the anosmia, we had what was that guy's name? Do you remember from Harvard who worked on anosmia?
It was Bob Data.
Bob Data. He showed that the virus reproduces in support cells of the olfactory neuroepithelium. So anyway, this mice, he infects them intranasally and they develop lung disease, as you might expect, right? The virus reproduces in the lung. But then if you let the mice go longer, you give them less virus so they don't die and let them live longer, the virus seems to go into the brain. It seems to be reproducing in the brain, which same with SARS-1, in fact. It
makes sense if you have the receptor there now in high levels, right?
Well, the virus has to get in, right, to the brain. It's not always easy. The brain is pretty well protected. But some viruses are very good at getting in, like poliovirus and enterovirus D68 and herpes viruses, et cetera. So just having the receptor isn't enough. But actually, that's a relevant point we'll get to. 5 of 8 mice are infected in the brain following 10 to the 5 PFU intranasally. And they can stain for viral antigens in the brain. They can see viral proteins, which suggests virus reproduction. And they say, the thalamus is affected in many COVID-19 patients with neurological disease. I still haven't seen good data for virus reproduction in humans. Have you, Ori, SARS-CoV-2?
There have been a couple of autopsy series, and I know that there's one from Colombia that's published. But these are, first of all, it's a weird population of people that have the disease because they're obviously people who had fatal disease. Also, many times they do the autopsies a while after the initial infection. But from what I understand, they see very little viral replication in the parenchyma.
I see.
And a lot of it is essentially qPCR positivity in low amounts, but in just tissue, ground up tissue. So it could also be from the blood, from the endothelial cells. But very little immunofluorescent data.
I mean, it could very well be that the neurological symptoms are immune response driven, right? So we don't know yet. But anyway, in these mice, the virus is clearly reproducing. And they say some of the affected areas are secondary or tertiary connections of the olfactory bulb, right? Which immediately suggests the virus gets in the olfactory epithelium and then goes into the brain that way, right?
Right.
Not everything. Some of the sites are not connected in the brain, but some of them are connected. So, they're thinking, well, in humans, we see anosmia, right? So let's look and see if these mice actually get infected in the sinonasal epithelium. And they do. They can infect mice intranasally. They can, of course, measure infectious virus in nasal secretions. But then when they look at the olfactory epithelium, they can see cell death. They can see viral antigen in sustentacular cells. I guess those are the support cells of the nasal, the sensory olfactory epithelium, right?
Right.
I think those are the ones that Bob Data said were infected in humans. So Bob said that probably causes the anosmia. So it's not infection of the neurons themselves, but the support cells. So then they say, do mice lose their sense of smell? And I read this in the title. I'm going, let me see how they get mice to smell.
Well,
how they
they're
measure
good at
it.
smelling.
How they measure it. So they have two.
Right.
This is typical. Now that I'm on TWiN, I understand these behavioral tests, right? These two tests require a sense of smell. And one of them is called the social scent discrimination assay. And they take male mice and they expose them to bedding that has either been on a male or a female mouse, or in a cage with a male or a female mice. And male mice like to lie in the bedding from female mice, okay? They say they preferentially spend time with female dander. It sounds very funny if olfaction is normal. And female mice are not attracted to male dander, which is kind of a nice control. And so mice with normal olfaction preferentially explore unfamiliar bedding, and they can see that when you infect them with SARS-CoV-2, the male mice don't go to the female dander. It's very interesting. The other test was a buried food test. Now, that's quite clear, right? You bury food and they have to smell it to find it.
Right.
And if you infect them with SARS-CoV-2, it takes them longer to find buried food. And the brains of these mice are not infected. They have minimal weight loss. Their mobility is normal, so that's not explaining why they can't find dander or cheese or whatever it is they're giving them. So it's the reproduction of the virus in the sustentacular cells is giving them what they call anosmia.
Right. Yeah, and that's consistent also with the fact that it's a transient anosmia. It comes back, and that's because those cells are, you can replenish them as opposed to the neurons. If you killed the neurons, most likely you wouldn't be able to
recover. Yeah.
Recover.
One interesting thing here is that, in mice that are sick in general perform worse on many of these tasks. As you can imagine, mice that are sick are not necessarily motivated to find food or motivated to find female dander. So it's hard to know whether this is true anosmia or another effect that is clouding our behavioral assessment.
Yeah, they say...
Yeah, that's a good point. And actually my colleague next door here at Utah, Matt Wakowiak, is one of the world's experts on olfaction in mice. And they've got some really sensitive assays where they can tell, whether the mouse smells one odor over another and that sort of things. There are sort of very physical kind of experiments you could do.
No, not based on behavior.
Not based on behavior. Yeah.
That's good. Yeah, that's good. Yeah, they say we don't think that it's generalized malaise or virus in the brain, but those experiments would be good to do. Anyway, that's really the part I liked about this paper. I think it's a model for anosmia, I think, and you can study. I'm not sure what you would do here because, once you have it, you have to just wait for the epithelium to recover. The virus is killing the cells, so it's not a really subtle effect, but it's nice to have an animal model, I guess, that recapitulates what's happening in people.
Right.
It would have been really cool to see if they had extended these experiments and seen if the anosmia, if these mice recovered, essentially.
Yeah, that would be good.
Do non-lethal infections, show anosmia, and then show recovery.
Yeah, right.
Would have added a lot of validity to the model. And then what do you think about the convalescent plasma data? So, essentially they show that convalescent plasma doesn't rescue the anosmia. And
but that makes sense, right? You kill the cells. Obviously, you're not. I don't understand how that would work unless you prevented the infection in the first place.
And they pre-treated the mice with convalescent plasma.
So they essentially treated.
They treated with convalescent plasma, then treated with the virus, and then
I see.
So it's. I mean, it's hard to explain, essentially.
Well, but I mean, those cells. So it's in the bloodstream, but is enough of that getting into the epithelial cells in the nose?
Well, I think the convalescent plasma prevents brain infection and lung infection, but it doesn't prevent upper tract infection. So why that is, it may be that not enough antibody comes out of because this is intravenously administered, right? Maybe not enough antibody gets out in the upper tract to protect them, or since you infect there, maybe it's just a big amount of virus and it's hard to overcome that. I don't know.
Yeah, or it's not IgA or something like that. Yeah.
I mean, there's some models, some non-human primate. I remember early on, one of the vaccine candidates was tested. I think it was an adenovirus vectored spike, and it protected non-human primates from lung infection, but not upper tract infection. So something's going on there. Maybe it's a similar mechanism. I mean, I think convalescent plasma we don't need anymore. Now, today Lilly's monoclonals got an emergency use authorization. Regeneron has an application in for EUA. They seem to work if you give them early. So if you have an at-risk patient, right, elderly or with some other health condition, and as soon as they have a positive test, if you give them monoclonals, it can work.
Right. So
or if you have someone in the emergency room who's exposed that you're really worried about, you can give it to them. The problem is that it's way more expensive than convalescent plasma.
That's true.
Where instead of having vats or having this production pipeline, you have people who are producing it.
The only problem is that Daniel Griffin points out the plasma has clotting factors in it, and
most people are hypercoagulable.
Yeah. So it may not be good. Whereas the monoclonals are purified. Yeah, but the government's paying for everyone's treatments, right?
Right. Clearly. I mean, that's the thing is the monoclonals are not cheap.
It could be 20, 30 grand for a treatment, right? Yeah.
Yeah.
Yeah, I know. That's true. But what was it? So if you give it early enough, it works. But if you give it in a patient who's already on oxygen, high flow oxygen, at that point, it's not a virus disease anymore.
Right.
Which we learned at some point. We didn't know early on, but it doesn't really help. Even remdesivir, an antiviral given late, doesn't do any good because it's not a viral-driven disease. Anyway. Okay. To wrap things up here, so we have one email from Margie, who felt bad that we didn't get emails, so she wrote like a 25-question email. So let's take one at a time per episode. We'll spread them out.
Sounds good.
All right. Greetings, TWiN friends. This is from Margie. I'm so excited to have a real reason to email you. I have been trying to come up with an excuse since TWiN 10 when you didn't have any letters and requested emails. Come to think, do I get bonus points for non-COVID-19 questions? Yeah. Background, I have multiple sclerosis, so a bit of familiarity with neurodegeneration. I was an elementary school teacher and then an elementary school librarian. I am now medically retired. Fair warning in advance. I am very good at unintentionally making short stories long. So apologies for the length. We're going to break it up, Margie, and spread it over multiple episodes. Today, Wednesday, 10/28, there was a report shared that caused quite a hopeful stir in my MS Gym Facebook group. Here's the link shared for the report made on a local TV news show. I'll also add screenshots of the transcript. So breakthrough research shows hope for reversing damage in neurological disease. Obviously, that report could be sensationalized, so I searched for more specifics. It turns out that the news is caused by a publication from a 26 October 2020 Nature Immunology title, A New Neutrophil Subset Promotes CNS Neuron Survival and Axon Regeneration. So this is a Nature Immunology. Yeah, here we go. Accept all cookies. Okay. Let me see the paper. Every website now has a big thing that says accept cookies, right? Oh, what a pain. Transected axons fail to regenerate, resulting in chronic neurological disability. In the current study, we identify a unique granulocyte subset that had neuroprotective properties and drove CNS axon regeneration in vivo, in part via secretion of a cocktail of growth factors. It promoted repair in the optic nerve and spinal cord. Have you seen this, Ori? No, or Jason?
No, I haven't seen this.
Okay, so Marge is suggesting we discuss this. So, okay, we'll put it on our list.
Mm-hmm.
All right, so one question. Oh, no, she's asking questions about the paper.
Maybe we can.
Let me see what this.
So the second, maybe we should answer the second question on this one.
Okay, second question. Both papers used mice. I've heard Vincent say mice lies, monkeys exaggerate, and ferrets aren't humans. So why bother with mice if they lie? This is asked tongue-in-cheek because I do understand that we have to start somewhere. Yet if mice and other animals are so different, structurally, molecularly, not sure the right words from humans, then what's the value of doing the experiments? That's not asked facetiously. I'd really like to understand. Okay. You guys, Ori, you can take a crack at that.
Well, I'll tell very quickly a funny story. In my thesis defense, someone in the audience asked what all my work meant and whether this could be helpful for disease. And I essentially said that it was just one long, crazy intellectual, stimulation project, and that didn't go over very well. So, I think that this mouse work sets the basis for understanding kind of basic mechanisms of biology, and then and testing hypotheses about what disease means. If you think about it, there are so many similarities. There's so much homology that even if it's not perfect, can obviously allow us to test how molecules work, how proteins work, how cells work, and even how something as complex as the brain works in an organism that we can relatively ethically study, that we can control for different variables, so we can have genetically homogeneous populations that we couldn't ever answer in humans. And I think that it essentially lets us generate hypotheses to go back. And, if you think about science, it's kind of one big circle where you see biomedical science. You see clinical observations that informs your ability to do bench research on models such as mice or monkeys or ferrets. And then you take those observations back to the clinic, either with treatments or to get more data to try to better understand the disease process. And we just have to start somewhere. And mice are kind of where we as a scientific enterprise have started because of ease of use and ease of manipulation. And I think we've made a lot of progress with them.
Yeah, I'll add that where the sort of disconnect happens eventually for translating science into therapies is at this level where you need a drug and the drug has to be safe, and the drug has to work like it does in the animals, in the animal models, as it does in humans. And so that's a big ask for a drug, but it doesn't mean that the biology is not conserved, right? It just means that to tinker with the biology is difficult. And I think that's where it gets lost, where we sort of complain about how mice aren't humans, but there's a lot more that's conserved than there is not. And certainly at the gene level and the biology level, at the building block level, it's the same. But it's how those things are arranged that's quite different. Of course, a mouse brain's much more small and it's tiny, and the immune system of a mouse is very different to the immune system in a human. But in terms of how they work in a coordinated fashion, but the building blocks are the same. So I think one could just think of this sort of building a recipe that has to be sort of slightly tinkered to help alleviate a disease, but at least gives you the ingredients to make the recipe.
Yeah, we say mice lie because you cannot use what you find in mice and say, it's the same in people. You have to find out, right? Often a drug or a vaccine that works in a mouse doesn't work in people. If it did, always we would just test the vaccine in mice and say, okay, give it to people, it's fine. But that's not the case. And this gene that was found in the study today that controls working memory, it might not control working memory in people. I mean, it's likely it does, but you can't assume that, right? You'd have to do some kind of experiments in people to show that. So that's what that means. You can't assume that whatever you find in mice is the same in people.
Right.
So that
Jim, I once heard Jim Allison say, so he's the guy who did a lot of work on kind of these immunomodulatory therapies in cancer. Yeah. He said that if you're a cancer researcher and you can't cure cancer in mice, then you're a pretty crappy cancer researcher. So in that sense, mice also lie, because you can say, oh, wow, this looks really promising.
Yeah.
And it doesn't work.
I also heard him say that, he found his T-cell therapy was working in mice. And he went to companies and they say, we don't care about mice, do it in people, and then we'll listen. Of course he did, and he got a Nobel Prize for it, and it's revolutionized some kinds of cancer treatment. But yeah, you start in mice.
Yeah, exactly.
And even if you try and get very close, if you want to use a non-human primate, the quickest thing comes to my mind, you infect a non-human primate with SARS-CoV-2, they get mild disease. They don't get the severe disease that some humans get. And why? They're fine. You put it right in their nose, it reproduces, it goes in their lung, they get a little pneumonitis, inflammation of the lung, but they don't get sick, they don't die. So right there, and they're 99% human at the genetic level. So, but we can't do experiments on people, we can only observe.
And of course, that's because the viruses are exquisitely evolved to specific species, and so
so the monkey environment is different enough so that even mice, these ACE2 mice, they die of brain disease, but they don't die of lung disease.
Actually we should talk about that. Well, I guess it's not a neuroscience but I'm fascinated by this observation in Denmark where SARS-CoV-2 has gone into the mink population, and then it's mutated in the mink population. Now it's gone back to humans. That is worrying. So it's also infected mink in Wisconsin and Utah now. It has, yeah.
And so whenever a virus goes into a new host, it will always diversify. It will become slightly different because there are different pressures, as we've been just saying, right? And from what I can tell from the Denmark, when they take those mink viruses, I think there are 5 amino acid changes in the spike. When they try and neutralize them with human convalescent sera, these are people that were infected with human isolates. They say there's a moderate reduction in the ability of the serum to neutralize. So I don't know I haven't seen the data, so I don't know what moderate means. It may not impact human disease whatsoever. We just don't know. So that's the concern, that, man, if these viruses were not neutralized by, say, vaccine-induced antibodies, that would be, to quote Amy, a shit show. Well, the whole thing is a shit show, right? But
Yeah.
So that's the concern.
We won't talk about the numbers. Yeah, Utah is a shit show right now.
But the last I heard last Friday from the Danish minister was that they said the virus hasn't been found in a month. This variant hasn't, or this isolate hasn't been found in a month. So I don't know what the situation is. I don't know if we have sequence from the American isolates either. So it's an evolving story.
Yeah, but they had to cull. I mean, they millions of animals. I mean, it was pretty dramatic.
Yeah. Well, the mink farms are tough because there are thousands and thousands of animals in close quarters, so it's really easy for viruses to spread.
I mean, I think personally, I think they shouldn't have had these farms anyway. But that's another.
Well, yeah,
question.
I mean, they're making them for the coats, and then they throw the west. It's kind of crazy.
How will we stay warm in the winter?
No.
It's well, they said in the Netherlands is getting rid of mink farming, and I think Denmark might. And then they say they have to retrain the farmers to do something else because this is what they've been doing for a living. It's very tough. It's very tough. Okay, that is TWiN 12, and you can find the show notes at microbe.tv/TWiN. And there you find links to the articles we talk about in our emails and so forth. If you want to send us a question, TWiN, TWiN, at microbe.tv. And if you like what we do, we have other podcasts besides TWiN. And if you like them all or some of them, you could support us. Microbe.tv/contribute. Ori Lieberman is at Columbia University Irving Medical Center. On Twitter, he's Ori Lieberman. Thank you, Ori.
Thank you, Vincent. Good to talk to you guys.
Are you still on psych these days?
No, I'm on surgery, which is almost worse. Yeah, but
surgery.
I've seen a lot of gallbladders removed in the last 2 weeks. So.
Okay, I guess you don't want to do surgery, right?
No, only one more week left, though.
Okay,
that'll be fine.
Jason Shepard's at the University of Utah, and on Twitter he is Jason Synaptic. Thank you, Jason.
Thanks, Vincent. I was going to ask you, did you give Nels a bit of treatment for this MacArthur Award he got?
Oh, I didn't. We didn't even mention it on the last TWiV. No, but he didn't tell me. I found out on the news.
Right.
Yeah, that's pretty good stuff.
Yeah. No, we're stoked for him.
Yeah, it's good. No, he's a very smart guy. We had him on TWiV last week to talk about his green. Do you know about his green zebrafish, Ori?
I heard a little bit on TWiV. I didn't finish the episode.
It's very cool. He put an interferon responsive promoter in front of GFP, made transgenic zebrafish, and when they get infected, they turn green.
Yeah, that's cool.
It's brilliant. And they found that their colony had a virus in it because the embryos were turning green spontaneously. It's very cool. Very good stuff. I'm Vincent Racaniello. You can find me at virology.blog. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back soon. See you next time.